After studying about numerical mutation, there comes another most important type of mutation, that is structural abnormalities in chromosome. Now, such an abnormality, whether in structure or number, is defined as chromosomal aberration. Or in precise terms, we can define chromosomal aberration as the changes that occurs in the number and arrangement of chromosome or gene can be defined as chromosomal aberration. Now, after studying about numerical change, let us start with the structural change. There are mainly 4 kinds of structural abnormalities or chromosomal aggregation, which includes deletion, second, duplication, third, inversion, and then comes the fourth, that is translocation. Thus, these are the 4 structural abnormalities that occurs within the chromosome causing mutation. This figure depicts the structural changes in chromosome. For example, the first one, that is deletion, in which a segment of a gene or more within a chromosome gets deleted. That creates a new gene segment. In the second type, that is inversion, the gene segment, whether 2 or 3, gets inverted at each other's position to create a new segment. The third is duplication. The term itself suggests that here either 1 or 2 gene segments within a chromosome gets duplicated. And finally, in translocation, part of gene segment that is present on 2 different chromosomes gets detached and joined to one another to create a new chromosome. So let us study each of them in brief. So let us begin with the first type of structural abnormality, that is deletion. This figure depicts the deletion type of structural abnormality within a chromosome. As it can be seen from the figure, this chromosome contains a DNA segment, that is gene segment, which breaks down, and this deleted genetic material gets separated to create a new type of chromosome. That leads to the structural change. Thus, deletion can be defined as the loss of 1 or more genes from a chromosome that occurs due to the loss of a chromosomal segment. Here, the genes are lost, and so the organism shows some defect. Now that is very obvious that if the normal chromosomal structure gets changed, then the functioning that responds to the normal chromosome gets also changed, which results into the defect. Such a loss can either from the end region or in between. That is, this deletion can either be terminal or inner region of chromosome. Depending on it, the deletion are of 2 types. That is, terminal and intercalary. Terminal can be defined as the deletion of a chromosome in which the genes are lost from the terminal region. That is from terminal part. The second is intercalary, That is, deletion occurs in which genes are lost from the inner region. This is all about deletion. The example of the diseases that occurs due to deletion of a chromosome includes various diseases in humans like Friedreich syndrome. It is a syndrome, or we can say the disorder or disease, that occurs in humans due to the deletion from the short arm of a chromosome number 5, that is short arm of chromosome number 5. That is, we can say that the deletion occurs from the autosomes. Following it, similarly, another type of syndrome or disease that occurs is Philadelphia syndrome. This type of syndrome occurs due to the deletion of the long arm of chromosome number 22. Now let us begin with the second type, that is duplication. This figure depicts the structural abnormality in form of duplication that occurs within the chromosome. As it can be seen from the figure that in such an abnormality, a part of the chromosome becomes twice, that changes the structure of chromosome and thereby its nature and characteristics. That is due to the replication of DNA causes a part of chromosome to occur twice. That is, we can say that it gets duplicated. In such a case, the sequence of genes is either maintained or get inverted. So, as it can be seen from the figure, in this duplication, the chromosome that contains a gene segment gets replicated or become twice and duplicated to create a duplicate genetic material and results into the formation of a chromosome that is entirely different from the normal one. Duplication plays a very important role in evolution, due to which new chromosomal structures can be introduced. Such a type of duplication generally occurs in Drosophila, where the abnormality in the size of the eyes occurs. And that occurs due to duplication. Owing to it, the repetition of genes occur and thereby additional characteristics are produced. The third type of structural abnormality in chromosome is inversion. This figure depicts the structural change in form of inversion. As it can be seen from the figure, the chromosome that contains DNA segment gets break down and such DNA segment gets entirely inverted or opposite to the original and then gets reinserted as a DNA piece within the same chromosome. Such an inversion also causes the structural change of the normal chromosome. Therefore, in inversion, a particular segment of a chromosome gets broken down or separated and gets reattached by getting inverted, that is, in an inverted position. Therefore, unlike deletion or duplication, here no loss or no gain of genes occur. But still, due to inversion, the sequence of the gene gets changed. However, sometimes due to this inversion, the phenotypic changes can also occur. Following it comes the fourth type of structural abnormality, that is translocation. This figure depicts the translocation type of structural abnormality. As it can be seen from the figure, here a part of the another chromosome segment gets separated, which joins with another chromosome segment, that is, both gets inverted and thereby translocated to form a chromosome pair which is not homologous to each other. Thus, in this manner, translocation can be defined as a structural abnormality in which a part of the chromosome gets separated and joins with another chromosome which does not result into the formation of a homologous chromosome. This type of translocation structural abnormality also plays very important role in evolution. Such a type of structural abnormality, that is chromosomal aberration, is generally observed in cancer cells. Now this is all about the structural abnormality in chromosome and its types.